Hi everyone, my name is Tara Popley and I'm a senior at Mission San Jose High School, and it is my pleasure to introduce Dr. Nazim Kavari and Dr. Nielsen Fernandez Becker to present on HB-2121, a new tool to study and treat celiac disease. Dr. Nazim Kavari is a pediatric gastroenterologist and integrally involved in the celiac disease program at Stanford University. Dr. Nielsen Fernandez Becker, an adult gastroenterologist and the medical director of the Adult Celiac Disease Program at Stanford University. And he also provides excellent care for celiac patients while also being the lead clinical investigator for multiple clinical trials, including the one that they're about to tell us about.
Hi there. It's so nice to see everybody here. My name is Nazim Kavari.
And I'm Nielsen Fernandez Becker.
Not be behind the screen. So I'm a pediatric GI. Dr. Fernandez Becker is an adult GI, and we've been working together for many years. And that's how we think we can provide the best care for our families and our children. So we want to tell you a little bit about one of the studies that we're working on, which is called HB-2121. And we'll start by just giving you a very basic outline of what we're going to cover over the next 15 minutes. We'll start with just a very quick overview of what celiac disease and why it happens. What are some of the unmet needs? And I know many of you probably know these quite well already. What non-dietary therapies are even out there? And then lastly, tell you about the study that we're working on. So what is celiac disease? You all know this already very well, but it's an autoimmune disease that affects primarily the gut, but also elsewhere. And it's caused by ingestion of gluten in a genetically susceptible individual. Many of you may have seen this diagram before, the celiac iceberg. And just like a regular iceberg, you can often see like the tip, right? And that's just a little bit of it, but actually there's so much about celiac that's underneath the water. And we can think about this in a lot of different ways. One of them is just in terms of like the clinical manifestations. So sort of the tip of the iceberg with celiac is stomach pain, bloating, vomiting, discomfort, right? And that's one that many people often recognize. But under the water, right, sometimes there can be much more subtle symptoms or even no symptoms at all. So I get headaches, I get joint pains, I get skin rashes, I feel kind of foggy in my brain, et cetera. And so when we think about celiac overall and sort of the clinical manifestations of it, it's often, pretty complicated and sometimes really, really easy to miss. I just mentioned, right? First, you need gluten ingestion for celiac to happen. You need to have genetic susceptibility. But having the gene, as probably many of you have heard, doesn't necessarily mean you have celiac, right? So about 30 to 40% of the population has celiac gene, right? Probably most of the people in this room. But then only about 1 to 3% actually have celiac itself. And so you need the ingestion of gluten, you need the gene, and then you need some sort of environmental factors, which we're still trying to understand, that seem to actually lead us to that next step of immune dysregulation. So here you can see one of our children consuming gluten, right? And then it leads to this immune response within the body. And then what ends up happening is the body actually kind of affects itself. And so the small intestine has kind of all of these little white blood cells that then affect the small intestine, lead to more cells in there, and then cytokines that not only affect the small intestine itself, but actually also probably go to elsewhere in the body. The way that we currently make the diagnosis of celiac is a combination of blood testing, which you guys are probably very familiar with, tissue transglutaminase when able, and sometimes some other antibodies as well, and then endoscopy, right? So then we try to take a camera, place it inside, as we were talking about, take little pieces of tissue to be able to look at it under the microscope and look for some of the characteristic findings of celiac. One of the things that's hard is celiac can be kind of patchy sometimes, right? So we do our best to try to get as many biopsies as we can, but sometimes it can take a little time to confirm the diagnosis. Our blood tests that we told you about are good, but they're not perfect. And then often the question comes, well, I've been on a gluten-free diet for a year. Is my intestine healed? And not everybody wants to put their children or themselves under another repeat biopsy to figure it out. Or you all probably know that in order for us to actually see the effects of celiac in the intestine, you have to be on gluten, right? And so often you're like, oh, I feel crummy when I eat gluten. I want to confirm my diagnosis, but I want to stop the gluten and it's going to take a little while. And you're telling me it takes 1 to 3 months that I have to be on the gluten. So then what? Like, do I let myself or my child suffer while I'm waiting to figure it out for sure? Or what do we do next? And so those are some of the places where our study kind of fits in. And I'm not going to go into too much detail on this, but there's a lot of complications with untreated celiac, and they range. And so, you know, one of the things that we're always thinking about is, are we treating our celiac well enough? We're thinking about quality of life as we're trying to be adherent to the gluten-free diet. And so, you know, right now, the repeat endoscopy is kind of one of the best tools that we have to take a look at that, or potentially the serology markers. But we're hoping that we can find other markers and other ways to do a better job, both in the diagnosis and then also in the ongoing evaluation of celiac.
Okay, so Dr. Kavari and I tag team in real life, and we're doing the same here. So in terms of the immunobiology of celiac disease, I'm not going to go through the details of this, but it's super complicated. Your small intestine is the biggest immune organ. There's lots of reactions that are going on. And what's happening is that the immune system should be able to distinguish what's bad for you and what's good for you. Protect you when there's something that is bad for you, like a virus, but not react when you consume food. And what's happening here is that gluten, for whatever reason, there's a lot of theories, is being recognized by the immune system as something foreign. And it basically sets off this cascade. Use this. Yeah, where gluten, after it's ingested, it goes into the compartment, yeah, into where the immune system is, which is just one single cell below. And all of these things happen. The really exciting thing right now is that in the table on the right is all of the different clinical trials that are going on. So basically, we're exploiting the immunobiology and trying to intervene at different points. And what we are doing is actually focusing on blocking something called, well, what you know as TTG or TG2. And why do we need to do this? Dr. Kavari already alluded to this, but the diagnosis that we make, it's not specific for celiac disease. Other conditions can cause the same degree of damage that we see. And the antibody tests are great screening tests, but they're not perfect to allow us to know how the intestine is healing. And we need better therapies because the gluten-free diet is not trivial and it doesn't work for everybody. So this is what, in essence, the clinical trial is. So the central part of celiac disease is that this enzyme, TTG or TG2, converts gluten that we ingest into a toxic form. So if you don't have TTG, then you don't get celiac disease. And we know from previous studies that one of the differences is that in people with celiac disease, this enzyme is always active. So what Dr. Chetan Khosla has done in his lab is develop a molecule that binds TG2. And it has a marker that allows us to see where it is. So, and the best part of it is that it's just restricted to the intestine. So this is a mouse model. And what you see, all this pink stuff, is this molecule. And it only stays in the small intestine, which is important because that will have less side effects. And then when you use a knockout mouse, which means that it doesn't have TTG in it, then we don't see any staining. And on the right what you see is the green stuff is all of the inactive tissue transglutaminase, or TG2. And on the, this one over here, the far right, is the molecule, it would come out as red when it binds to TG2, but red and green make yellow, so that's what you're seeing. So it allows us to pick up the disease. So in somebody who has active celiac disease, it lights up. But because of the way it works, it is also a potential treatment. So right now, we just completed the first phase. So in clinical trials, the first thing you want to do is to prove that it's safe. So we tried it on people who don't have celiac disease and people who had well-controlled celiac disease, and we found no safety concerns. Now we're on to phase 1B, which is what we're going to tell you about, where the idea here is anybody who's undergoing an endoscopy for the purposes of evaluating for celiac disease, we're trying to prove that this improves the diagnosis by providing a specific marker. And so we're looking at safety. We're looking at also what's happening in the lining. And we're also looking at the blood to see if we could pick up any markers that are non-invasive so that we don't have to do an endoscopy every time we want to evaluate. So all of this, and we're using a dose of 250 milligrams because that's what we found works the best in the previous study. And we're looking for 20 participants. And this is, sorry, this is our team. I'm the principal investigator, and Dr. Kavari and Dr. Yo are sub-investigators. And this is something that has been funded by Stanford. And these are our clinical coordinators. And now I'm going to pass it on to Dr. Kavari.
And I'm noticing that we're running low on time. So I'm gonna maybe skip this one. So the medication is a powder, and we basically put it in a bottle of water. And it's a bright blue because that's kind of the fluorescent stain that helps light up. It's what we call open label, so everybody knows that they're getting it. And then basically we do this is for anybody who's ever been interested in clinical trials or just kind of curious about what it looks like. We thought it might be interesting for you all to see. So basically what we do is first we do what's called informed consent. So anytime somebody's involved in a study, we say, hey, these are the risks, these are the benefits, and this is kind of what it looks like. We do a screening visit. The day of the routine endoscopy, you come in, you get to drink the liquid, we do the endoscopy, and then we do follow-up visits over the course of the next month with lots of blood testing, urine testing, et cetera, just to make sure that it continues to be as safe as we found so far. Oh, I'm frozen. Okay. One of the questions that people kind of always ask are clinical trials safe? And so just for sort of everybody's knowledge, we have multiple practices to help make sure that things are safe. We have institutional review boards, so they're scientists and others who are completely not at all invested in the study, who take a look at our data and who take a look at everything every so often after every so many participants to make sure that what we're seeing as safe is also what they're seeing as safe, if there's any additional testing that needs to be done or other interventions. All of that said, right, every trial, anytime you try something new, there's risk and there's benefit. And so as we kind of discussed earlier, probably the biggest risk is just that it's still very early testing in people. So far, we really haven't seen any negative and we really think that this is really sticking to the gut only, but we are still in process of always learning and checking. The big side effect we've seen is that the poop and the pee turn bluish green, which is related to the dye that we just gave them, right? The benefits are, it helps us, hopefully help improve the diagnosis of celiac, maybe help us find a biomarker that we can follow in the long run. And then also hopefully sometime be able to make the diagnosis of celiac without putting people back on gluten. And then eventually maybe treatment.
Yeah, so what happens afterwards is we analyze all the data, and this is under the auspices of the FDA. And hopefully, if everything works out, the next phase would be to try to do a bigger trial that we could really tease out safety. But I couldn't be more appreciative of all of the patients who have participated in all of our different trials. The organoids wouldn't have been possible. Nothing, and this is a human disease, so definitely we need human studies to advance. So thank you very much for considering it.
Can I just add an extra thank you to all the people who have allowed us to take a few extra biopsies or a little extra blood, because if it wasn't for that, whether it's pediatrics or adults, we couldn't do it. So just to add to that again.
Hi, my name is Emma, and I am 15 years old, and I am a ninth grader at American High School in Fremont. And it is my pleasure to introduce Dr. Vanessa L. Kamari, a physician scientist at Stanford University, who has dedicated her work to celiac disease research. She has been looking into new ways to measure the intestinal damage and inflammation caused by celiac disease using a swallowable capsule that can sample the small intestine without need of endoscopic procedures. Today, she will share how this technology is opening new doors for celiac disease research and what it could mean for patients like you. Please join me in welcoming Dr. El Kamari.
Thank you so much, Emma. Hi, everyone. I am very excited to be here. So my name is Vanessa Al-Kamari. I am an infectious disease doctor. You would ask yourself probably, why is an infectious disease doctor doing here? Because of an interest in understanding how initially different viruses and bacteria could affect our gut immunity, I got involved with celiac disease about 3 years ago. And I fell in love, really. It's such a wonderful community. And I have since dedicated my research to it. And I really hope to dedicate the rest of my career to it. Wonderful community, the best of the... So not going to go over all of this. I'm sure you guys already know, like, what are the biggest challenges that we still face? And a lot of it have already been, like, Dr. Javari and Dr. Fernandez Becker have, like, already touched about, is, like, really, like, some of the struggles in being able to non-invasively monitor celiac disease. The small intestine is really a place is where the disease happen. And one of the biggest issues, and it's one of the most important organs in our body. But at the same time, it's one of the hardest organs to reach. It's really where all of the nutrient absorption happen. About 70% of our immune system lies there. And we really have no idea of the microbes and the microbiome that are there. Usually, whenever we want to study that, we will rely on stool samples. And as you can all remember from your biology classes, I'm sure, the stool is really very far from the small intestine. So whatever you're getting from the stool is really very different from the environment that you see in your small intestine. And kind of like we said here, currently the only things that are available to access the small intestine is endoscopy, which I'm sure is not a lot of fun for a lot of you guys know here, and then blood, which is not a best way to predict what's happening inside of the gut. So this is why we have been collaborating with a biotech. And together we have been developing this wonderful device called the CapScan device. And what it does is that it allows us to sample some of the small intestine in a non-invasive way. So this is the capsule that the device that you actually, okay, so this is the capsule that you actually take. It's the size of a vitamin pill. And inside of this capsule, there's a small tube that is usually just embedded in it. And the way that it works is that the capsule is designed in a way that whenever it reaches a certain pH, the membrane of the capsule will just disintegrate. And inside, the tip of the capsule will show up. And as it's traveling through the GI tract, it will keep opening and it will start sampling some of your intestinal lumen fluid. So basically, you just swallow it, it's a pill. And based on the pH, it's designed in a way it will just disintegrate and then start sampling. And eventually, you just catch it in your stool, such as a tube. And I usually always carry it with me. I forgot today, but it's really the size of a pill. And then the tube is a very flexible, not without any it doesn't have any electronics or anything. It's really a hollow tube. It's just made of a one-way valve that allows the fluid to just go and fill the whole tube as it's traveling. So we end up having, this is a real sample that came out, and this is how it would look like. So you would have different segments. They're not segments, it's a one tube, but since it's just like a one-way valve, what you end up having is like different fluids from along your GI tract. And we can clearly know that those are from different parts of the intestine. And what we end up doing is that we cut up this tube. We usually get about 600 microliter of fluid inside. And with all of the technology today, that is enough to give us a lot of information. So we will cut it up and we will put it in a small tube that will go for testing. So what do we measure? A lot of things. Among them, I'm not going to go into all the details, but we're able to measure basically all of get the DNA test of all of the microbes that live there, about the millions and millions of microbes that live there. We're able to measure a lot of the enzymes that exist there, which can try to help us differentiate whether there are some patients who have some enzymes that other people don't have. We're able to measure some of the cytokine, which is basically like the surrogate for inflammation that is there. So in other words, we're able to directly have a readout on what is happening in the small intestine, the site where celiac disease is. So the study we are undergoing, you guys have met most of the team. You're aware about most of the team here. I couldn't have done this without the great collaboration of our physicians here. Inflame study is the name of our study. We are recruiting adolescents 14 years and older. We're also recruiting adults. This is the age that we think the capsule so far has been safe for, but we hope that there's a different design of the capsule that is smaller that we think that will come up and could help with smaller kids. And the people we're recruiting are patients or participants that are newly diagnosed or with celiac disease, or the one who are on untreated celiac disease, who have been taking gluten and are about to start a gluten-free diet. And what we do is basically they take the capsule along a year. It's up to them. It depends really on each participant, how much they want to get involved, how much they have time to get involved. But we basically, the goal is to be able to monitor how they do over time, up to 1 year after starting their gluten-free diet. And at the same time, to understand what a healthy gut looks like, we have other healthy controls that we're also recruiting. And in the past, I want to say month or so, we have added a new group of patients that we have been looking at. And those are participants who have been with celiac, who have been on gluten-free diet for the past year, but still have some symptoms. And as you can imagine, the goal is really to try and understand why some of those patients, despite gluten, are still having symptoms, and trying to get a better sense of their intestine milieu. We are short on time, but basically, if anyone is interested in participating, it really depends if you're an adult or a kid. For adults, kids, adolescents at this time. It's pretty straightforward. We send the kit at home, and everything can be done at home. If you are between 14 and 18, it requires to have one clinic visit, pretty easy. It takes about 45 minutes. And what we do is you'll just swallow the pill. We'll take some blood. We take some saliva, and pretty much it. Ask you some questions, and then we'll ask you to just collect those, collect like the capsule and at home, and then send it back to us. We use a pretty cool device for the blood, which is amazing. We should all be doing this. It's called the Tasso kit. This is a very non-invasive way to get blood. No needles, nothing like that. We just put it here and doesn't hurt and just gives you blood. So far we have had in our study alone about 60 capsules from celiac patients have been taken. Across different studies, this is happening in different disease too. We have about a couple of hundreds of patients have taken this capsule. So far, we've had no complication. It works great. We have, from our preliminary studies, we've been able to see that it really is very sensitive to show us things that we can see in blood. And then we have been also working and collaborating with different departments at Stanford, where we have been able to develop some probes that have been able to show us very sensitive way how we can track inflammation over time. Obviously, there's a lot we can do, and we hope to do with that. Really, the big goal would be eventually to find this is just a different approach, a different way that could help us clinically, but also could help our patients every day in monitoring their disease. But at the same time, also give us a lot of insight about some of the mechanism of celiac disease that we still don't know, including how is the different environment affecting, and why are some people with celiac still have some symptoms? How are the microbe affecting that, among a lot of others? So please let us know if you are interested in any way to get involved. And thank you for everyone who have made this possible, especially our participants who have been wonderful so far, and all of their families. Thank you so much.